Okay, the meninges are the protection of the brain and spinal cord, or offer a tremendous amount of protection to the brain and spinal cord. So we have three of these covering. Collectively, we call them the meninges. The outermost layer, or the superficial layer, is called the dura mater. The middle layer is called the arachnoid mater. And the pia mater is the innermost layer that actually touches the brain and spinal cord, following every fold, almost like it's shrink wrapped very tightly around the brain and spinal cord. Okay, now as you're seeing here, we're looking at, so here's the scalp, the skin, hair. Next layer in is periosteum of bone and the bone itself. And then now we're getting into the dura. And then you're seeing the arachnoid mater, which is the purple. And then you see this space. There's a particular space there called the subarachnoid space, and that is where cerebrospinal fluid circulates. And the light blue that you see there, sky blue, that would be the pia. So that's the pia mater. Okay. All right, cerebrospinal fluid begins in the choroid plexus. Okay, so this is a specialized group of blood vessels that extract fluid from the blood. So the choroid plexus is a specialized group of blood vessels that extracts fluid and makes cerebrospinal fluid. Okay, so that happens in the choroid plexus of the lateral ventricle. And then it is released into circulation. You can see the arrows, so it spills into the third ventricle, and then eventually through the cerebral aqueduct and into the fourth ventricle. Will work its way through the cerebellum, down the spinal cord all the way down to S2, S3, and then circulates back up and around the brain just to start all over again. Okay. To zoom in tight, you're seeing the choroid plexus, the ependymal cells that we talked about in an earlier lecture, and as fluid is being extracted from the blood, it will enter into brain circulation through the ventricles. Okay, this is out of Netter, which is a pretty detailed anatomy book, and you're seeing here that they're peeling back the dura mater, so here it is covering the spinal cord, and seeing the arachnoid, and then there's a cut there, and then you're looking at the pia. Okay. Also take note of the gray matter here. They've recessed a lot of the white matter to have the gray matter stand out pretty aggressively there. So this is the length of the cord. So the gray matter and white matter is the entire length of the cord. Okay, meningitis, so that word itis means inflammation of, and it's inflammation of whatever word it's attached to, so in this case the meninges. So meningitis is inflammation of the meninges. Virus and or bacteria can cause this, and they typically do a lumbar tap or a lumbar puncture, which is a method of extracting cerebrospinal fluid to analyze the CSF, see what's going on. Okay, the CSF is made from blood plasma, has less protein than blood, does tend to have some vitamin C in it, and it is continually made from the blood by the choroid plexus, of which we looked at earlier. The plexus is a cluster of capillaries within that ventricle in the brain, and that would have been the lateral ventricle. And the CSF aids in protecting the brain and spinal cord via a water cushion. Okay, now on this diagram, we mapped it out through those arrows on the picture, but this is the written version where the CSF is continually moving through the lateral ventricles and into the third, through the cerebral aqueduct, and into the midbrain, into the fourth ventricle. And then here the fluid will go into the cerebellum area and down the spinal cord through the central canal. All of this occurs in the subarachnoid space. Okay, it's important to know the CSF circulates in the subarachnoid space, and the fluid will return to the blood and the dural sinuses through the arachnoid villi. And the average adult has about 150 milliliters that will circulate. Okay, if there's any abnormalities like meningitis or any other condition, lumbar tap is performed where they'll look at color, they look for blood or the presence of blood, if they find different types of proteins in there, or any abnormality. After the lumbar puncture and the extraction of CSF, the patient will usually be required to lay down for a period of 6 to 12 hours to avoid a headache or other complications, because once you remove CSF and you go from, say, 150 milliliters down to, say, 145, well, that loss can cause a significant headache. So that's why it's advised to stay recumbent for 6 to 12 hours. As far as technique is concerned, the anesthesiologist likes to go in at the level of L3. So if you're noticing over here, there's your needle that is going in, and it is going into the subarachnoid space. Okay, now remember, the conus medullaris, the spinal cord, goes from foramen magnum down to L1, L2. Okay, and the end of the cord is called the conus medullaris, and the continuation of it is the cauda equina. So the reason why they like to go in below the level of L3, as you're seeing there, is because there's more room for error. You're actually away from the spinal cord, and if you hit anything, if you go deep enough, you're hitting cauda equina stuff. Okay, so there's more room for error. They're in the subarachnoid space, and they're going to extract the fluid. Okay, they like to have you in a fetal position or in flexion. That flares the spinuses, gives them more room to go in.